Okay, so I think with that, I will start to move on to the next part. So in this part, we want to look at two notebooks. And the idea here is I just want to sort of give some idea of accessing the archive. So you grabbed an archive entry, and how do I access the metadata, the data and the metadata in Python? And so a lot of this is sort of going to be very basic, like how do I access the data? And then I'm going to show you one tool which we implemented, which we can then expand to other transformations in the future, which converts the archive entry into a structure of a third-party analyzer, which then you can do like some standard analysis with ND trajectories. Okay, so in this first part, I'm going to look at this is like some analysis of a protein solvation structure. And so this first part is going to involve this system that I introduced before. So this thing's in my way now. Sorry. So it's this protein that's solvated in water, and this protein is classified as a certain domain type, which is called PDZ2. And these domains exhibit allosteric behavior. So basically, when a molecule comes and binds to this protein, it has some functional change. This often occurs with some structural change of the protein. And so what people might want to do, so here's a reference if you're interested of like where this data comes from. And what you might want to do is to run simulations, molecular dynamic simulations of this protein with and without this ligand, and then look at what aspects of this ligand affect the structure, or what role, for example, water plays in this ligand binding. And so we're going to look at some analysis of this protein without the ligand. So just to mention a few things about, so we have this container you can use, but there's not so much that's going on here. I am using a couple modules like directly from Nomad, so you would need to install Nomad locally if you wanted to do this locally. Otherwise, they're relatively basic requirements, including this MD analysis package, which is this third-party analysis software that I'm going to use towards the end. So in general, you can use the Nomad API. There's a link here to grab particular archive entries from the repository. And the only reason I didn't do that here was because I just wanted to supply the data directly to you for simplicity. And so here I've just pre-downloaded this entry, and I've stored it in this JSON file, similar to what we uploaded in tutorial 2. By the way, I already pre-executed these cells because I had to make sure that this would load. Yes.
Joe, we are not seeing the notebook.
Oh my gosh.
We are still seeing the overview, yeah.
Thank you. Well, that's
Thanks, Sasha.
Yes. I mean, that's weird. It's on my desktop. You see it now?
Yes, now we see your voice code.
Okay,
Yes.
Sorry, I don't know. I guess I clicked browser before. Okay, so I was just talking about these. So again, this is the beginning of the notebook, part 1. Here's a link to the reference, and here I was just talking about the inputs here. And then, no, I think I was down here. Yeah, I was just saying that. So I provided this JSON file, and I've already executed most of these cells because I wanted to make sure everything loaded in time and I didn't have issues. So this is already loaded. And so when I load this data structure so I'm just reading a JSON file, and this is a dictionary. And if I look at the keys of this dictionary I'm doing here, then I can see that these are the sections that we investigated before when we were looking at this data tab. So this is directly corresponds to what you can click around in that data tab on in one of these entry pages. So we have this run, for example, and this workflow section, etc. So what I want to do first is convert this dictionary into the internal Nomad entry format. And so you can also just simply work with this dictionary. I think once you get used to it, the archive format is like slightly more convenient to work with. But I also need it in this case because I'm going to call this function which transforms this archive entry into a different data structure. So by the way, these times that I put are, I guess, probably an underestimate depending on, so this was on my laptop when I wasn't running Zoom, and you can see that like it took me a lot longer. So it just depends on your system, but it gives you some idea. Okay. So the first thing we can do is define some sections. So again, this section run, this is, I can define this as the archive and then put dot run. And then there's actually, if you look at, so I'm gonna make this archive.run. So I can see this is a list. And so there's only one entry, so there's only one run in this list. So I can equivalently put -1 or 0, and that will be my section run. And then section system. So this is similarly just, again, like going through this data tab. So section system is section run dot system. And then this, I'm going to call this section topology, system topology. This is this atoms group section that we looked at before. This had like the groups of molecules and such. And this was stored, if you remember, in this first configuration in the system section. And then I have section atoms. And here I'm just defining this as, again, for the first configuration. So I'll loop through the different configurations later. But this is just to grab some basic information about the number of atoms and such. Oh, yes, there possibly is a question. Or people are just talking. Okay, people are just talking. Someone let me know if there's a question. Okay, so here I just wanted to show, okay, we can go into this archive and we can pull out the relevant information. So let's say we want to analyze the positions and velocities, and we should get the box vectors as well because we want to apply periodic boundary conditions for our analysis. So that this cell would do that. So I define the number of atoms. This I can get directly from this section atoms, and I can use .git on this, and just to ensure that this is well defined. And then the number of frames is just the length of this section system. And then the atom names I can get, it has an attribute, section atoms has an attribute called labels, and this I'll get the list of atom names. And then I can go through and get the atom positions, velocities, and box dimensions. So here I'm going to predefine the length of these arrays, just so we're clear. So the shape of this positions is the number of frames by the number of atoms by 3, and similar for the velocities. And then the box dimensions are just going to be the number of frames by 6. So the first 3, I'm going to store it like the first 3 are going to be the box lengths, and then the second 3 are going to be the angles, and they're just going to be 90 because this is a cubic box. And so now I can loop through the system. So for each element in this list, so if you remember, section system is a list, and I can loop through that, and then I can get the corresponding atom section. So I'm looping through frames, and I'm doing frame.get. So this gets the atom section. And then under the atom section, I have an attribute called positions. So I get that position from this positions frame, is section atoms frame.positions. And then what I can do, so I'm going to use this package called ureg, which in this case, I imported from the Nomad module. And I'm going to use this to convert the units of my position. So the quantities in the archive have units, if applicable. So the positions have some units, for example, SI units. And here I want to convert these to something more convenient to work with. So I'm going to convert them to angstrom. So I can use this function ureg.convert. The magnitude of the positions I get by taking the positions for this frame and then doing dot magnitude. And then I can get the units by taking the positions for this frame dot units. And this is going to convert these positions then to angstroms. So that's this is u8.angstroms. And I'll do the same thing for the velocities. And then the lattice vectors, these are found in section atoms. So this is for this particular frame, and then dot lattice vectors, or I'm using dot get here. And then again, I'm defining some conversion factor, and then I'm going to convert those, and I'm going to store these in this dimension. So I'm not actually going to use this. I'm just showing you how you would, so you can look a little bit more closely, how you would access these quantities and then convert them to some to some different units, and then you could use them for some analysis. So let's do something a little bit easier though, in terms of just plotting something from the archive. So here I sort of left it as an exercise in the notebook, so I'll just sort of walk through and type these in, and then you can think about them. So here I want to define section run again. So I did that above actually already, but let's do it again. So section run, it's under the archive, right? So it should be archive.run, and then there's only one in this list. So we can use also, maybe we want to investigate sometimes these things. Okay, so that's that. And then section system is a subsection of run. I'm going to keep going, but please someone tell me if there's a relevant question. So this is section run.calculation. Again, you can sort of find this out by either exploring in your notebook, or you can go to the data tab of sort of a corresponding entry, and you could sort of click around and see what's there. Okay, so I want to find now the temperature unit. So if I go to section, let me actually define these above. So I have section calculation. Let's look at what is inside of here. Okay, so I have a list of calculations that should have been expected. We saw before there's a list. So let's just look inside of one of them. So inside of one of them, we have these attributes, and there's one attribute, temperature. So I just want to look at this temperature. So this is just for one configuration. So again, this is where I have so this quantity has a magnitude and a unit. So I just want to get the units for this, assuming they're all the same. And so here I can then just do, so it's section calculation, and then I'm just going to take the first one, and then I will want to grab the temperature, and then I just want the unit. So the way to grab here, I can either type magnitude or units, and then I'll get the corresponding quantity. So Kelvin. Okay, so that's how I get the units. And similarly, if I go back to here, there's another quantity, which is the time in calculation. Go up, so there's time, and then I can do the same thing. So this would be like section, whoops, calcu. Okay, it's helping me. I'm just going to take the first one again, and then time dot units. Okay, and then now I want to loop over each member of this calculation list, and then I want to grab the temperature. And here I just want to store the magnitude in Kelvin. So I'll put calc dot temperature dot magnitude. Okay, and then similarly for the time, I want to store calc dot time dot magnitude. Okay, and hopefully I've typed everything correctly, and I should get. Okay, I have now this trajectory of the temperature as a function of time, and this one is what exactly is displayed as I showed you before in the overview page under thermodynamic properties. There was one of them was the temperature. Okay, so in the next part, I did something a little bit more complicated, or at least I looked deeper into the sort of archive, and I asked we should pull out one of these RDFs that were plotted in the overview page. And so here I've already typed in the answers, but basically, again, if we go to workflow and there's just one entry in this workflow section, and then molecular dynamics. This is that section I showed you in the data tab, which had all the molecular dynamics information. And then I want to go to results, and under results, you'll find a section which is called radial distribution functions. And how this is organized is that this radial distribution function section holds all radial distribution functions of a particular type, or some group of radial distribution functions. So let me show you actually. So I will define this ND section, and then if I just look at this, there's actually only one. Sorry, that's not what I wanted to do. So if I look at section ND dot radial distribution functions, there's just a list, and then there's one section which is radial distribution function. And if I select that one and then do type, this says molecular. So we've left it open that you could also calculate atomic radial distribution functions and then place them in this section. So for the future, when things the user can supply particular radial distribution functions that are of interest. But for now, these are the pre-computed ones. And then if I go to, so these are all the molecular radial distribution functions. And then if I go to this subsection, which is radial distribution function values, I have now a list of a bunch of radial distribution functions. And so if I take the first one and I say label, label, it's going to tell me this is a radial distribution function between the protein chain, so the center of mass, and the sodium ion, the counter ions. And then I can see that there's also the start and end of the averaging. So remember, we calculated the radial distribution function for different sections, and so these values are stored to tell you how we're averaging. So in this case, I just want to grab the very last one, which is the solvent-solvent, so the water-water radial distribution function, and it's the longest one. So it's I'm actually not sure if it's the longest one, but it's it may be the last 40%. I forget now. But anyway, so you can grab that, and then we want to define the start and end here. I'm going to use this later to reproduce this RDF. Okay, so I can execute this, so it should be fine. So this is the radial distribution function. This is one of them, again, that will be displayed in the overview page. Just want to briefly check the time. Okay. So now what I can do is I can convert this archive instead. So let me say, so what I typed here, it's somewhat clear that, okay, we can go into the archive as we did here, and we can grab all the positions and velocities and whatnot, or whatever quantities we're interested in, and then we could do some trajectory analysis. But here, then we'd have to implement the periodic boundary conditions, and depending on what we want to calculate, there's a lot of tools out there to already do this. And so we want to be able to convert very efficiently into a format that we can apply sort of modules that already exist. And so one way to do this would be to use the MD analysis module. And so there's already a function implemented in Nomad that we use to convert this archive entry into what's called a universe. This is just the data structure that MD analysis uses. So I've done that. And what I can do then is use the sort of various selection tools and such that are pretty advanced in MD analysis to start to perform some more detailed analysis. So first of all, I can just look at the molecule types so if I do universe.atoms.mol types, then I'll get all the molecule types for each of the atoms. And if I do np.unique, then I get the different molecule types. So there's 3 different types. I'm not going to go into great detail about how MD analysis works, but I'm just going to try to give you an idea of what you could do once you with this archive data. So what I'm going to do first is I'm going to reproduce this RDF, that I calculated above. So I take my universe. This is just this trans-converted archive data. I'm going to select all the atoms which are solvent, and I make an atom. I can make an atom group out of that. That's what I've done here. And because I'm calculating this, because the Nomad in this overview page is showing the center of mass radial distribution functions, this is actually somewhat complicated, like non-trivial to do with to do center of mass conversions with MD analysis. So I'm using here some idea, some function which calculates on the fly the center of mass. So let's not worry about that. But anyway, I need this to define this bead group. So it's just transforming these solvent molecules to the center of mass essentially. And then I'm going to define some parameters like the box dimension and the maximum RDF that we want to calculate and the number of bins and how we want to smooth this thing. These I'm just choosing as the same as what Nomad is using so that we get the same answer if we do this properly. So these are just sort of input parameters. This did not execute. Okay, well done. That's unfortunate. Okay, so we'll wait. Oh, I don't know if that may be a problem. Actually, oh man, can I not? Can I just stop this? Does anyone know in VS Code if I can just stop the cell without restarting the kernel? I don't know.
Between beside code and markdown, there's interrupt. You go up.
This one.
Yes, this one. I think this one will.
But it won't break my. It won't break it, right?
I think so.
I think so too, but I'm a little bit nervous because I want to be able to.
Maybe we can also wait.
I think there's also an interrupt next to the cell. Right next to it, there's a small stop.
This one?
No, okay, never mind.
Yeah, I think okay, we give it a sec. We give it like a minute and just to see, and I'll keep talking about this. So anyway, basically, I mean, it's not even important. I should have executed it. Basically, I'm just showing, okay, if I can use this like built-in function in MD analysis, that's this iter RDF to calculate the RDFs, and then okay, I get the same answer, fine. But that's sort of the basic idea of how I would like take this archive data and then okay, thanks. Okay. So then again, so I can use this sort of selection ideas from this MD analysis tool now, and I can so I can do like universe.select protein. This will get all the protein atoms, and then I can grab information like from the topology essentially. So like this just says, okay, there's 83 residues in the protein. And then I want to do some more like specific analysis for this system. So it's known that the first lysine and first phenylalanine residues, they make hydrogen bonds with like a ligand that comes and binds this. And so I want to select those residues, and then I want to do some analysis. So that's what I've done here. So I'm selecting here all atoms that are involved in lysine, and then I can actually look for just the unique residue type, residue IDs. So the residues are numbered, and then I'm just going to pull out the first one. So that what I'm printing here is like, this is all of the residue IDs that were found. And as I said, it's the first one in the chain. So I'm just going to extract this one and call it the binding pocket residue for lysine. And similarly for this other residue, phenylalanine, there's two of them. And I'm just going to pull out the first one. Okay. And then what we can do if this is almost finished. So it'll take 2 minutes, but I think it'll take more like 4 minutes, maybe even longer. Okay, let's just keep. I'm gonna keep going, and then I'll come back to this. So this is just wrapping the coordinates for visualization, and then I have, if you click, so if this is live for you, you can click through this. Basically, you'll be able to view the system as I showed you in the sort of Nomad overview page. But then you can like highlight sort of easily the protein, and then you can highlight these residues that we picked out. So here I made them like bigger than the other proteins, and then I can go further and actually do like more complex selection commands. Like I can ask to, within this one configuration, to select waters that are like 3 angstroms, 3 nanometers, or I'm not sure what I'm using here, from the residue, and yeah, from the residue, which is this lysine and the one in the binding pocket. And then you can see that these waters are sort of in the solvation shell of this lysine and this phenylalanine. And so hopefully I'll be able to show that in a second. But then, okay, we can do some other like standard analysis, like perform some hydrogen bonding analysis. So I don't really care. I don't really want to explain all the syntax, but this is again just some empty analysis thing. So there's built-in analysis to do hydrogen bonding computations. And so if you run this, then you can make a plot like this, which says this is as a function of time, the number of hydrogen bonds with these binding pocket residues that we selected. And here we're looking at the hydrogen bonds between so I selected here the oxygen as the donor atom and the hydrogens as the water hydrogens as the the opposite, but that's what this is the acceptor, and then the donor is the hydrogens of the water. And here we can see that we have like some hydrogen bonds with phenylalanine, but not with lysine. And
Joe, it's the square, just not the arrow next to it, and then you stop it, and it works like just a keyboard.
Yes. Okay.
Interrupt.
Which next to here?
Yeah, just a square next to the. I mean, right where.
This one.
To the left. No, to the left of the cell. Just at the.
Oh, I don't see it. I don't see it because I didn't see it. Okay.
And then it works like a keyboard interrupt for just that.
Thank you. My Zoom was in the way. I can see that. Okay, great. Thank you very much. Okay, let's see if we can. I have to run this. See if I can view this. Sorry, everything's running a bit slower on my computer. I'll give it 20 seconds, and if not, I'll just move on. Any questions so far? That's a good. Doesn't seem to be the case. Okay, maybe I'll just move on, and if this ever wants to execute, I'll come back and show you. Basically, basically, if you click through these, you'll see that there's water molecules that are around the lysine, and there doesn't appear to be very many water molecules around the phenylalanine, which is exactly the opposite of what we saw in this hydrogen bonding analysis. So it's a little bit misleading, but if you look closer, you can see that actually the oxygen of the lysine is, oh, it worked, okay. So let's just view this, and then I'll explain what I had in mind. Oh, wow. Okay, interesting. Okay, so here I'm just viewing all the system in like the simplest way possible. And then I can execute, so this is NGL view that I'm using, and so I can highlight the protein. And then I can look at the specific, so these are the binding pocket residues that we, oh, they're not defined. Okay, anyway, we can highlight those. So this is not that important. Let me just explain again, sorry. So if you follow these, then so there's this water around the lysine, but they're not very close to the oxygen. The oxygen's like buried in this protein. whereas for the phenylalanine, even though the whole residue is somewhat buried, the oxygen is pointing out into the solvent, and that is consistent with the analysis we did below. Anyway, the point is that with these we can do this analysis by grabbing an entry from the archive, and we can also use these tools to convert the archive entry then into this different format. And these converters possibly we could explore depending on what people are using this data for. Then we could make other converters to sort of interoperate between different modules. Okay, close that. I'm also going to close this because my computer is now very low on memory. Okay, so let's move to the second part. And in this part, we're going to look at a polymer simulation. And this is recent work by Banerjee et al. So there's a link here that you can see the work. And the data I'm using is not from this work. It's from a follow-up work, which is not yet published, but the idea of the analysis that you can find here. So the basic idea is to try to identify the glass transition from some features of individual polymer chains. Again, for the imports, we're going to use similar to what before. We're going to use this MD analysis package, and then this converter function from Nomad, and then I'm going to use some other basic sklearn functionalities. Okay, so I preloaded this data. This takes quite a long time to load, and what I'm doing here then from this data is, sorry, I'm going to go up. So I already loaded this data. I converted it.
Joe?
Yes.
So there is actually a question regarding the post-processing from Jan Lian. So he's asking, can FAIRmat only does post-processing analysis, isn't it? Can we use it to extend molecular dynamics calculations using all the parameters we uploaded?
Yeah. Great question. So yes, so far we've been we're mostly focused on storing the data, right, and then making it accessible to others, and then the next step would be exactly what so the next step would be what I'm showing you. You can do some analysis on an existing simulation. That's easier. I can just grab the positions, velocities, and whatever. But then sort of a next step or a couple steps further would be what you're saying. So can I grab this entry and extend the simulation? And for that, you would, that's much more complicated. You'd really need all the relevant parameters, and we have most of them, or as many as that are available within these standard outputs. But I mean, it's quite an undertaking basically to take those and then put them back in the simulation package. There's actually some other projects which are focused on that aspect in a way, not coming from Nomad, but they're focused on how can I run simulations in a sort of fully scriptable way, and how can I interoperate between different codes. And if you combine sort of both of our approaches, so we're sort of taking the end product and going back and storing it, and they're sort of trying to go forward. And then there's the possibility to connect those two things, and then we could try to do at least approximately what you're saying. I mean, approximately in the sense that you could start simulations from configurations that exist. I mean, completely reproducing a molecular dynamics simulation is much more complicated if you're trying to use a different software, but that's a whole nother topic. And so this is exactly what we're looking into, is how can we make connections between various consortia which are focused on different aspects of this problem. And in fact, there will be, I'll give a little advertisement, there will be a CCAM meeting in Mainz next September in person about this topic, where we'll bring together a bunch of people in this community who are working on these projects. And in the near future, on February 27th and 28th, there'll be a small workshop, virtual workshop about this. So you could hear people talk about these different projects if you're interested. And perhaps Lucia could post the registration link for that workshop in the chat if you're listening. Otherwise, I can post it at the end. Okay, I hope that answered your question. If not, please add more in the chat. Okay, so I was saying that here I am loading the data from these pre-parsed archive entries, or these. So these are the archive entries, sorry, that were downloaded for this data. So I have a series of temperatures, simulations at a bunch of different temperatures. These are polymer melts. Thank you, Lucian. And I'm first loading them from the JSON format. Then I'm going to convert each of them to these archive type format. And then I'm going to just go ahead and convert them to this MD analysis universe format as I did before. Was there something? Okay, so that I already did. And then I'm going to show you, hopefully the visualization works this time. I'm going to show you the system and such, but what I'm going to do is I'm going to take one of the chains and then I'm going to featureize the configuration essentially. So I want to eventually compute pairwise distances along this chain. And I'm going to select some atoms to do this between. So I'm going to select these sort of mainly side chain atoms and two on the backbone. Okay, so that I did here. And again, I can just use these selection commands from based on these universe. So I select so these are the names of my atom types. And then, this is just making a string of like my selection string. This is just like the syntax in MD analysis. And I'm going to go through each of the simulations. So each of these different temperatures, I'm going to select out atoms that are part of a single chain. So I'm just going to look at the first chain in the system, but there's many chains, so you could do this for each one. And then I'm going to also make sure that it's in this molecule, and then it's one of these atom types, basically. So I'm going to select all these atom types within a single molecule. And then I'm going to, so now I want to just visualize the system. So I already did this unwrapping of the coordinates, and let's see if we can make this work. My computer is slowly dying. Okay. There we go. Okay, so it's a pretty big system. So this is the whole system. And now I can look at just the first chain, which is what I'm going to do the analysis on. Okay, so here's the chain. I think I can, yeah. Okay, so here's the first chain. This is what one of the polymers looks like. And now I can select, I can view just the selections that I made. So all the atoms in blue are part of my selection. So I'm just selecting these carbon atoms, and that's going to be part of my analysis. And then what I'm going to do is I just want to calculate all the pairwise distances between these, excluding the self distance, right? And so I'm going to make essentially like a matrix out of this. So I'm just going to loop through my temperatures, and then I can use this. This is a function from MD analysis that I imported. So I'm just going to calculate the distances for this entire group. So what this is calculating all possible pairwise distances within a single group, which is I defined in these atom groups, and I'm doing it for each temperature. So I'm grabbing the positions here. And then what I'm going to do is just stack them on top of each other so that it doesn't matter which temperature they came from. I just have one long array, or I have a matrix of the configurations, regardless of the temperature, and then of each of these features, so each of the pairwise distances. So that's what I have in distances here. Sorry, this is still separated by temperature. I apologize. And then here I'm stacking them together. And when I stack them together, I'm going to make this array of temperature labels so that I know which temperature each configuration came from. And then what I want to do is I'm just going to do a simple dimensionality reduction of these features with principal component analysis. So just a linear dimensionality reduction. So that's what I'm doing here. So I'm just pre-processing the data so that they have a 0 mean. And then I'm just going to plot here the first two dimensions of the principal component analysis, a scatter plot of all the configurations. And now I'm labeling the configurations by color based on the temperature, as you can see in the legend. And here we clearly have, so we have some separation in the configurations based on if they're like low temperature configurations or high temperature configurations. And I can go further by doing a clustering of this data. And so that's what I'm doing here. And then now I'm going to plot just the cluster label. And basically, you can make some separation that these are so low temperature and these are higher temperature. And this transition interface between the two, from this, you can approximate what the glass transition temperature is. And this is sort of much more. So if you go back to that reference from Banerjee et al., you can see that this is much more precise than sort of the typical way you try to look at the estimate the glass transition temperature from sort of these like phased transition plots. So I guess the volume as a function of temperature. Yeah, and so also here, I just wanted to thank Autry for providing this data and this analysis for us to look at today. So yeah, so that's the end of that. And again, this is just to give you some idea of what you might be able to do if we, by grabbing molecular dynamics data from the Nomad repository and then processing it in some systematic way. And this sort of motivates trying to get users to upload this type of data. And we hope that now that we have support, so I didn't mention it explicitly, but there's now 2 types of codes that are supported, so GROMACS and LAMPS. And we'll now begin to try to expand that a bit to some of the more flexible codes, and also to work on some of these metadata aspects. Like storing the force field and being able to easily grab this data and convert it, and hopefully eventually, even as was suggested by the question, be able to provide tools to easily spawn simulations, although that's not our focus, but with collaborations with other consortia.